going to continue in the short-term tradition of black background slides, which seems to be a trend here. So I'm going to tell you about some ideas I've been developing recently as part of my dissertation work. But before I do that, I would briefly like to thank my advisor for Denison, who you can see is outstanding in his field, and my committee, funding sources, and Will Radcliffe, a former graduate student in our lab whose dissertation work really formed the foundation for some of what I will be talking about today. So, imagine a horizontally transmitted symbiont outside of its host. Not a lot of resources there, not many opportunities for reproduction. Then, one day, it encounters a host and enters the host, and then, thanks to the rich supply of resources it encounters, the host manages to reproduce and exit the symbiosis with a net fitness benefit. So, what often happens, though, is that within the host there is constrained reproduction for whatever reason. For example, there might be an imbalance of resources or limited space. So what a lot of bacteria do when there's an imbalance of resources, an excess of some and a shortage of others, is that they store the excess resources in various polymers inside of their cells. So then the fitness benefit of symbiosis is not just how much they manage to reproduce, but also how much they manage to sequester, which may confer some type of fitness benefit in the nutrient-poor environment in the soil or the air or what have you. So my dissertation research focuses on a particular type of stored resource that's common in bacteria. It's known as polyhydroxybutyrate, or PHB, which always trips me up because I say I'm getting a PhD in PHB and it gets confusing. So you can see PHB granules in these little white globules inside bacteria cells. And you may have actually heard of PHB in the context of biodegradable plastic. It's actually being investigated as that, but it also pops up in a wide variety of symbioses. So, for example, Legionella bacteria, responsible for Legionnaire's disease, they accumulate PHB inside of amoeba hosts, and this may contribute to their ability to persist in nutrient-poor environments such as air conditioning systems. PHB also pops up in some marine bioluminescent symbioses. So the light-emitting bacteria that live in light organs of various marine fish and squid, you can see sometimes they get PHB granules that could contribute to their persistence in the water. And of course, PHB shows up in the legume rhizobia symbiosis, such as soybean and alfalfa. Here you can see a cross-section of a soybean nodule with really, really, really big PHB granules. In fact, measurements of soybean nodules have found that PHB can make up 50% or even 70% of bacterial biomass within the nodule. So that seems like a pretty big cost from the perspective of the host plant. And that's actually what got me interested in PHB in the first place. So nitrogen fixation and PHB accumulation both use the same metabolic precursors in the TCA cycle. So from a game theoretical point of view, I thought, okay, nitrogen fixation, that's kind of like a public good. It benefits the host plant, helps it grow. The host plant then can photosynthesize more, provide more carbon, benefits all of the bacteria. But PHB is more of a private good. That's just the individual bacteria cell accumulates this resource, potentially helps its fitness outside of the host. But this potentially word is where I kept getting tripped up because I realized we really don't have a good understanding of how PHB acquired during symbiosis contributes to fitness after symbiosis. So that's really the question I've been spending most of my time on. How does PHB affect fitness between hosts? What's the relative fitness of, say, a high PHB strain versus a low PHB strain, and how do you even conceptualize that problem? So, to give you a little context, I'd like you to imagine a typical soybean-corn crop rotation. And this might be easier to imagine if you're from the Midwest, because they're everywhere. But once the nodules from the soybean plant senesce at the end of the season, they release a population of rhizobia. Some will have PHB. And then they will need to make it through a winter, a year of corn, and another winter before there's another opportunity for nodulation. However, even when a new nodule comes along, a fairly small fraction of the rhizobia in the soil will actually make it into a nodule. So much of this original population will need to persist even longer, perhaps to the next soybean crop, perhaps indefinitely. So the question is, how does PHB contribute to the ability to persist for long periods in the soil? So I thought of two possibilities. The first is that PHB sequestered in hosts provides a short-term fitness advantage. So strains with more PHB have a slight boost in reproduction shortly after they're released from nodules, and then their descendants have a better chance of either making it into a nodule, winning that reproductive lottery, or persisting in the soil thereafter. But there's also another possibility, which is that instead of reproducing, those cells go into a dormant state and use their PHB slowly for long-term survival. So they themselves might have a chance of reaching a nodule or surviving longer in the soil. So these two hypotheses I'm calling the frugal use hypothesis, so using resources slowly for long-term survival, and the prodigal use hypothesis, using resources quickly for short-term fitness. This type of trade-off might be familiar to those of you who study, say, annual or perennial plants. So the first question in considering these hypotheses is how long could a rhizobia cell survive in the soil on stored PHB? So is it even biologically plausible that a cell, say in a dormant state of metabolism, could survive until the next soybean crop or even longer? So to answer this question, or explore this question rather, I did a little physiological modeling of carbon metabolism. So I found some published rates of temperature-dependent carbon metabolism for a wide variety of bacteria from various environmental sources that were extrapolated by Price and colleagues. The blue line is from Price's work, and that was for bacteria that are active but not dividing. So they call this maintenance metabolism. So everything, all systems are go except for growth, which requires external resources. And then on the other end of the spectrum is dormant metabolism. So this is the minimal level of metabolic investment necessary to repair DNA, reverse the switch from T to D amino acids, basically maintain long-term viability, but without doing anything. So I also considered some intermediate rates because these published rates came from not rhizobia. And since metabolism is temperature dependent, I've plugged in some realistic soil temperature data from a field site I've worked in Waseca, Minnesota. That's southern Minnesota, typical soybean growing region, and just fitted a simple temperature oscillation curve. And so once I plug in soil temperature data to metabolic rate, I get approximate range of how long a rhizobia cell could survive on the PHB it gets from hosts. So here in the x-axis is a range of PHB per cell that comes from measurements I've made in soybean nodules. So that's kind of a biologically plausible starting point. And then the dotted lines are my rates of dormant metabolism, and then the solid lines are active metabolism. And you might notice this oscillation here. That just comes from seasonal changes in temperature, metabolism slowing down during the winter. And something that's pretty interesting is that let's just look at a cell that starts with 0.5 picograms. So it could survive for less than a month, or it could survive for over 100 years. So that's kind of a wide range. And it might seem like, an individual cell surviving for decades or more, that, might seem far-fetched. However, in the agronomic literature, there are many examples of symbiotic rhizobia populations maintaining at sticking around at relatively stable levels for decades. In a couple of these cases, these two were soybean nodulating rhizobium introduced to soil that lacked soybean nodulating rhizobium and then recovered many years later with no detectable changes in phenotype. So I'm not saying that all of these are survivors of what was initially put there, but some of them could be. So next, I wanted to see how rhizobia use their PHB when they're starving. So I got a set of rhizobial isolates from Waseca, Minnesota, isolated from soybean nodules. I inoculated them onto soybean plants in growth pouches, and then harvested their nodules, made a delicious nodule slurry, diluted it in starvation buffer, and placed it in these alloplots, some of which I still have in my lab, and just have been collecting them over time. And then I measured PHB with flow cytometry. So raise your hand if you've used flow cytometry data. Okay, good, there's a couple of you. So, audience specific. So flow cytometer, what it does is it measures light properties of very, very small particles. So what I have here is a population of rhizobia extracted from nodules. And I can quantify PHB fluorescence with a quantitative lipid stain, such as mal red. This is just showing microscopy images of 2 rhizobia cells of PHB. And then I can also get a proxy of cell size, which I'm not using, but basically this demonstrates that bigger cells tend to have more PHB. So one interesting thing I saw within the first week of starvation is that I got this separation into high and low PHB subgroups, which if I gated these 2 populations, estimated their PHB using standards, I found that the low PHB subgroup always had approximately 0 PHB, whereas the high PHB subgroup had more. So that's actually what I used to estimate, to observe PHB use over time. So what happened? This just shows a representative sample. So the change in daily PHB use rate in picograms per day over time. And then the solid lines here are my model parameters for model estimated PHB use rates. So it's nice when your data fit with your model. But what happened was that within the first 30 days, the metabolic rate slowed dramatically, going from the active range to the dormant range. And this happened in pretty much all my rhizobia strains. Here's another one just to demonstrate. And since I'm almost out of time, let's see, a couple of minutes. So another interesting pattern I saw was that high PHB strains use more PHB in the first month. So each of these is, each dot is a different rhizobia strain, and how much PHB they started with predicted how much they used. So this suggests that prodigal use, using resources quickly for short-term fitness, may be an important function of PHB. However, I also found that strains that started with more PHB also brought more PHB with them when they went into dormant metabolism. So it suggests that frugal use may play a role as well. So I presented these as 2 different hypotheses, but what really probably happens is some type of mixed strategy where perhaps some individuals are using up their PHB and some are using it slowly, or so group level mixed strategy, or an individual level mixed strategy where an individual will use a portion of its PHB for short-term fitness and then a portion for long-term survival. So I mentioned this separation into high and low PHB groups. I still have a minute. So what I think happens is that these high PHB group is actually the parent cells, old pole cells that have kept their PHB from the nodule, whereas the low PHB group are the offspring. And we actually have evidence of this from soybean nodulating rhizobia and alfalfa nodulating rhizobia, that the old pole cells keep all of their PHB and they get this asymmetric cell division. So in summary, rhizobia could survive for many years on stored PHB, and that there might be some sort of mixed strategy of short-term prodigal use and long-term frugal use, potentially at individual or group level. So that's what I have. Thank you.